Thank you.
Thank you. So since I'm the only thing between you and dinner, then we'll try and get through this quickly. And the good news is that there's no discussion. So I just need to tell you that I consult to anybody who would listen, and I take money from whoever will give. So lucky for me, Burton and his colleagues have actually produced a table which actually gives us a very nice approach to identification and characterization of genetic determinants of complex disease, of which psoriasis and psoriatic arthritis are. And so the first thing that we have to do is we look at the recurrence risk ratios, and basically the question is there evidence of phenotypic aggregation within families? Before we do that, we just want to talk about the phenotype of psoriatic arthritis. Now, we've heard earlier from Philip H. and Philip M. that about the various patterns of psoriatic arthritis. But if you look at psoriatic arthritis as an entity, we recognize that there are several domains, and they include the skin and nails, peripheral arthritis, axial arthritis, enthesitis, and dactylitis. And so when we talk about the genetics, we need to consider all these various components of the disease. Now, when you look at family-based investigations, we've known for many, many years that there are genetic factors in psoriatic disease, and those are based on the idea of clustering of both psoriasis and psoriatic arthritis in families. And in fact, if you go to any series of patients with psoriatic arthritis or any series of patients with psoriasis, you find that if you ask them, do you have a family history of psoriasis or psoriatic arthritis, 40 to 50% of the patients will say yes. And the majority are going to have psoriasis, but about 15% are going to have psoriatic arthritis in a first-degree relative. And it's also been known for many years that there is a high concordance rate among monozygotic twins. And the actual evidence comes from epidemiological studies that revealed a recurrence rate of affected siblings, which is the lambda S, of 8.2, and there are two different studies in the 60s, and there are a number of studies that looked at the concordance for psoriasis in monozygotic twins. It was almost 70% compared to 20 to 23% in dizygotic twins. And there's a recent study from Denmark that actually showed the same thing. Now, what about family investigations? So John Mall, together with the late Werner Wright, actually did a study where they looked at first and second degree relatives of 88 patients with psoriatic arthritis that were seen in the clinic, and they sampled consecutively and unselectively 181 first degree relatives. 10 had psoriatic arthritis, and 5 of those were actually siblings. They calculated, therefore, that the prevalence of psoriatic arthritis among these first-degree relatives was 5.5%, and taking the prevalence of psoriatic arthritis in the UK in the early 70s as 0.1%, they calculated a risk recurrence of 55, which is quite high. In our center, Vinod Chandran has actually looked at 100 consecutive families of probands with psoriatic arthritis, and you can see that there are 289 first-degree relatives, 130 were siblings, and looking at the number of people that had psoriatic arthritis, there were 22 for a prevalence of 7.6%. Those with psoriasis only, there were 44 with 15.2%, and calculating the lambda, this is for first-degree relative, was actually 30 for psoriatic arthritis, and very similar to what we saw in the earlier studies for psoriasis at 7.6. When you look at siblings, again, it's the same lambda for psoriatic arthritis and similar lambda S for siblings for psoriasis. And this is assuming a prevalence of psoriatic arthritis of 0.25% and of psoriasis of 2%, which is what has been recorded for North America. So we know that there is a genetic predisposition. The question is there evidence of a gene with substantial enough effect to justify extensive studies to attempt to identify it? And the answer is yes. The major histocompatibility complex, which is on the 6th chromosome right here with class 1, class 2, and class 3 components, has actually been identified as a major site of genetic contribution to disease. So this is a recent study that was done in New York, but the study population was psoriatic arthritis and psoriasis patients from Dublin. And what you see here, and this is looking at psoriasis compared to controls, they have a discovery subcohort and a validation subcohort, and this is the combined cohort. And what you can see is that the HLA-B57, whether it's alone or in combination, is a haplotype, and the HLA-B37, again, alone or in a haplotype with CW6, or B27 was were found to be associated with psoriasis, but this was not shown in the combined cohort. And when you look down here, there's B18 and B40 and B8 and C7. So a number of alleles. Now, this study only looked at class 1 alleles. They didn't look at class 2 alleles. Again, in our center, Vinod Chandran did this as part of his PhD thesis, and he looked at 684 patients with psoriatic arthritis, 706 healthy controlled of self-reported European ethnicity, and we identified the ethnic outliers using principal component analysis, using appropriate SNPs across the genome, and looking specifically at these areas. And as a result of this, we had to exclude a few patients with psoriatic arthritis and a few controls that turned out not to be quite ethnically European. But these are the results. In the univariate analysis, C1, C2, and C6 were associated with psoriatic arthritis. C12 was also associated with psoriatic arthritis in the univariate analysis, as was B27, B38, and B57 compared to healthy controls. And then a multivariate analysis retained the B13, B27, B38, B39, B57, and DRB101 as associated with psoriatic arthritis. I should mention that these were confirmed, at least the ones that are highlighted were confirmed by a family investigation. So this is the HLA class 1 alleles. If you look at TNF alpha polymorphisms, the 238A variant has been associated with psoriatic arthritis, and this is a meta-analysis that was done looking at a number of studies confirming this association. Now, we've also looked at MIC-A, which is the major histocompatibility related chain, and what we found was that if you look at psoriatic disease versus control, this is including patients that have psoriasis alone and patients with psoriatic arthritis, you can see that there is an association with a haplotype that includes the C6, B13, and the MIC-A008, as well as the C12, B38, and the MIC-A002. If you look at psoriatic arthritis versus controls, you find that it's a slightly different haplotype frequency. And when you look at psoriasis versus control, you find that the 0657, 017, and the 0613, 008 are associated with psoriasis compared to controls, and the same one that's associated with psoriatic arthritis. So there are haplotypes that are associated with the disease, both in terms of psoriasis alone and in terms of psoriatic arthritis. We've also identified an IL-1 gene cluster in psoriatic arthritis, looking at 212 patients and 150 controls, and you can see that there are a number of markers within the IL-1 gene cluster that are associated with the disease. And of course, IL-1 is on a totally different chromosome than the ones I've just been talking about. We've also looked at killer immunoglobulin-related genes, KIRs, and in an older study that we did with Mary Carrington at the NIH, we identified that B27 and C6 were associated with psoriatic arthritis, as a dominant model, but the KIR2DS1 and KIR2DS2 were associated with psoriatic arthritis independently. In a more recent study, we were only able to confirm the KIR2DS2 with an odds ratio of 1.25. Now, based on the model, one could actually describe an association with disease between the KIR allele and the HLA allele, because the KIR ligand is the HLA molecule, CW6. And so if the patient has both the KIR and the ligand, they are protected. But if they have the ligand without the if they have the KIR without the appropriate ligand, then they're much more susceptible to having the disease. So there's definitely evidence of genes that are substantial enough to proceed with these kind of investigations. So where in the genome are those genes? Well, we have a number of susceptibility loci that have been identified for psoriasis. So there's SORS1 on chromosome 6, and it's thought that it's particularly the HLA-C6. The SORS2 is on chromosome 17q, and then a number of potential genes in that area. SORS3 is on 4q, and it's the RIF2. SORS4 is on 1q21, and again, there are a number of possible genes. SORS5 is on 3q21, again with a number of genes. SORS6 is JUNB, and that was actually identified initially from a mouse model. SORS7 is on 1p, the short arm of chromosome 1, and it's got the IL-23, which we've heard about before. SORS8 is on 16q, and again, a number of important immune mediators. And there is SORS9 on 4q and SORS10 on 18. So there are a number of chromosomes and a number of loci within those chromosomes that are particularly important. So can we be more precise about the position of the chromosomes? The answer is yes. So in red, as you can see here, are the locations on the chromosomes of psoriasis susceptibility genes. And in green, as you can see here, are psoriatic arthritis susceptibility genes. And in yellow are those that have been identified for both psoriasis and psoriatic arthritis, and you can see that they occur in quite a number of chromosomes. Now, recently, there have been a number of genome-wide association studies that have been done, and this is one that we've been associated with validation, and you can see that this is the first study was done in Germany, and there's another study from Germany, another study which was North American, and another study which was British, and this is the combined association, and you can see that a number of genes have been associated, the IL-12B, I have to look in here, RYR2, NF-kappa-B, and TRAF3IP2, so a number of genes that have been associated. This is particularly for psoriasis. And there was a meta-analysis that was done of the immunochip testing, and this is a Manhattan plot, again showing that the major association is with the major histocompatibility complex on chromosome 6, but you can see everything above the green line are genes that have been associated with psoriasis, and some of these, of course, are also associated with psoriatic arthritis. And again, the genes that are associated with the areas that have been identified are very much immune-mediated genes. And these are new loci that were identified through the meta-analysis when it was possible to combine a number of studies. Now, importantly, those genes are relevant to the pathogenesis of both psoriasis and psoriatic arthritis. So this is looking at the skin, and this I received from J.T. Elder. So there is the skin, and there are the cells that are relevant in the dermis. And what you can see is that many of the genes that have been identified have skin barrier functions. And then another series of genes that have been identified have an innate immune response function. And then additional genes that are in blue are adaptive immune response. So the genes that have been identified for psoriasis and psoriatic arthritis susceptibility are very much immune-mediated and very relevant to the function of the skin. And as I mentioned earlier on, many of these are also very functional in the joints. So the question that still remains is, does the polymorphism affect messenger RNA? In which tissues is it expressed? And does it actually translate into an effect on protein product? And the answer is yes. So this is biomarkers looking at the protein products in patients with psoriatic disease. So this again combines patients that have psoriasis without arthritis and patients that have psoriatic arthritis. And if you look only at the multivariate analysis, you can see that there are 4 biomarkers: the rank ligand, the TNF-SF14, matrix metalloproteinase 3, and COMP, which is a cartilage biomarker, are associated with psoriatic disease. There's a study that was done by JT's group where they looked at microarray gene expression of 58 paired lesional and uninvolved psoriatic skin samples and 64 control 6 samples. And you can see that there is quite a difference between the psoriatic skin and the normal skin. And look at the number of expression markers that were identified within these samples. Now, this is only in the skin. And we think that some of these are also going to be in the joints. Now, this is a study that we did in Toronto where we took 20 patients that have psoriatic arthritis and 20 patients that have psoriasis, which we labeled as PSC, means that they only have cutaneous psoriasis. They've been assessed to assure that they don't have psoriatic arthritis. And again, you can see that there is a differential expression of microarrays between these patients, and they were matched on age, gender, psoriasis duration to make sure that the people with psoriasis without arthritis had enough of a chance to develop psoriatic arthritis, and also on the severity of their psoriasis. We identified 98 differentially expressed genes. So in summary, there are very strong genetic components for both psoriasis and psoriatic arthritis. And many susceptibility genes that have been identified are related to innate immunity as well as adaptive immunity. We're still working on genetic factors that can identify psoriasis patients that are destined to develop psoriatic arthritis, and I'll get into some of that tomorrow when I talk about the risk of developing psoriatic arthritis among patients with psoriasis. Thank you very much for your attention.